Eu vou passar agora para falar em inglês, né? Porque nós temos uma convidada, Simona Stäger, do Canadá. Hi, everyone. I'm very glad to introduce Simona Stäger here at Fiocruz Bahia. It's not her first time at our institute. In 2011 in person, she was here and gave us a fantastic talk. Simona is a professor at the Institute Armand-Frappier Research Center since 2011 in Canada, Montreal. She made a very interesting postdoctoral fellowship at the Imperial College of London, of Science, Technology and Medicine at London, as well as in the School of Hygiene and Tropical Medicine. She was assistant professor at the Johns Hopkins School of Medicine for a couple of years. And then she moved to Canada, where she is from 2011, a professor there. Her background, interest in veterinary, and her main interest in immunology of leishmania infections. And then in this field, she gave important contributions on CD8 cell biology, mainly in the development of effector and memory CD8 T cells, as well as in the priming of CD8 T cell response. We are very glad to have you here, Simona. Many thanks to have accepted Juliana's invitation to be in our seminar at Fiocruz here. It's a very honor. Thank you very much.
Okay, sorry, there was, can you see?
Yes, we can see everything. It's okay.
Mute. Okay, I don't know why it goes back to mute. Okay, so thank you very much, Patricia, for the kind introduction. And let me put the pointer. Voila. So for the kind introduction, and also thank you to Juliana for inviting me to give a seminar at your institution. And it's a pity that I cannot be there in person, because I really always love to come to Salvador and have the great food you have there. So like Patricia said, today I'm going to give an overview of what we have been doing in the past few years in my lab on the regulation of T cell responses, CD4 and CD8 T cell responses during experimental visceral leishmaniasis. So as I've seen in the crowd, you have a lot of leishmaniacs, so I don't need to give you an introduction about that, because you definitely know better than me about the disease. So I'm just going to go very, very quickly. There are different clinical forms. We work with the visceral form of the disease, and which can be caused by Leishmania chagassi infantum and Leishmania donovani, and we work with Leishmania donovani. And this is the worst clinical form, because it's actually fatal if patients are not treated. So symptoms are hepatosplenomegaly, hypergammaglobulinemia, fever, cachexia, anemia, pancytopenia, immunosuppression, and as I said, if people are not treated, they die. This parasite is transmitted by sandfly vectors, and which this sandfly regurgitate the mastigote form, the flagellated form of the parasite, into the skin of the host. And in the case of visceral leishmaniasis, this parasite is visceralizing within 24 to 48 hours. And then it develops into a chronic infection. So the main cells that are infected are macrophages, but you can also find Leishmania amastigotes inside neutrophils, dendritic cells, and in some cases fibroblasts and monocytes. So we work with a mouse model of the disease, and it's not a natural model because we couldn't get it to visceralize if we infect in the skin. So we inject amastigotes into the mice IV. This model was developed by Jenny Blackwell at the London School of Hygiene and Tropical Medicine, and I learned this in the laboratory of Paul Kay, that is still using this model. The mice also develop hepatosplenomegaly, but the liver and the spleen and the bone marrow behave very differently. So in the liver, the infection is actually peaking at day 21 post-infection, and then is 21, 28 post-infection, and then is kind of self-resolving. And this is thanks to the generation of a granulomatous inflammatory response, which on one side it contains the infection, on the other side it recreates the environment necessary for killing the parasite. So both CD4 and CD8 T cells are required. So if you deplete either of them, you get an exacerbation of disease, or if you infect granzyme B knockout mice, you get an exacerbation of disease in the liver. And these T cell responses are initiated by IL-12, which is also an essential cytokine. They produce interferon gamma and TNF, which activates the infected phagocytes, mainly Kupffer cells and macrophages, to produce nitric oxide and intermediate oxygen radicals, which kill then the leishmania. So we are not so much interested in the liver, because it's more a model for a subclinical visceral leishmaniasis, and we are more interested in the spleen, that is more a model for chronic visceral leishmaniasis. And in the spleen, and like in the bone marrow, the infection behaves very differently. So basically, during the first 7 days of infection, you hardly see any increase in the parasite burden. And then between day 7 and 14, you have a slow increase in the parasite burden. Between day 14 and 21, all of a sudden, there is a disruption of the microarchitecture of the spleen, and the parasites start multiplying dramatically. There is also emergency hematopoiesis that occurs during this time, and there is an in-stream of myeloid cells, neutrophils, and inflammatory monocytes. Mainly, and an onset of a chronic inflammatory environment as well. So it's in this chronic inflammatory environment, leishmania loves this chronic inflammatory environment, divides very happily, and there are lots of these monocytes that arrive that are really permissive to infection, like safe houses, and so the parasite establishes a chronic infection. So this goes along as well with an enlargement of the organ, which is a splenomegaly. What is interesting is that the splenic architecture, the disruption of the splenic architecture, is mediated by TNF. It's a cytokine that is actually required in the liver to control the parasite burden, but in the spleen it does a lot of damages. It also recalls IL-10 responses. So in the absence of TNF, you have less IL-10 in the spleen, and you don't have the disruption of the splenic microarchitecture. So this is the environment in which T-cell responses are supposed to be primed and maintained and be effective as well. To display their function. So how they are regulated in this environment. And I'm going to divide my talk into two parts. The first one is the regulation of CD8 T cell responses, and the second part on the CD4 T cell responses. So CD8 T cell responses, what I like best, actually, but unfortunately in leishmania, they are not doing much. So we, in leishmania donovani, to study the CD8 T cell responses, we had to use somehow artificial model, because there are no known epitopes for CD8 T cells. So we use the ovalbumin expressing amastigotes. These were generated when I was still a postdoc in Paul Kay's lab. And if you want to use them, you should contact him, because there is an MTA, so I cannot give it to you. And these parasites are actually producing ovalbumin. They are not expressing ovalbumin, so they are producing ovalbumin on the surface of the membrane. And this allowed us to use DOT1 mice, which have CD8 T cells that are TCR transgenic and recognize the synthetic peptide of albumin. So we developed a very simple approach. These are all data, so I'm going to go pretty quickly. A very simple approach. We transfer CD8 T cells the day before the infection into congenic mice, and then infect with amastigotes, of as transgenic amastigotes, and then we sacrifice the mice at different time point to monitor the development of these responses. And we could see actually that the CD8 T cells undergo clonal expansion and then clonal contraction. And then they stay more or less on the same numbers until day 30, and all of a sudden they disappear. But what is interesting is that this expansion is actually very limited. And if at peak expansion, so around day 9, we have 0.1 to 0.2% of the CD8 that are OT1. And if you compare that to what has been published for malaria sporozoites expressing Synfecal, so with the same method and the system and the same amount of cells that were transferred, so we transferred 20,000 cells, the percentage for malaria is about, for falciparum sporozoites, are about 1%. So that means that is that's like a 50% reduction in the expansion of leishmania compared to another parasite. And if you compare it to virus, it's even more than that, even greater than that, the limitation of the expansion in leishmania. So that means they expand, but very poorly. And during this clonal expansion, only 10% of the cells are actually short-lived effector cells, so express PD1, KLRG1, and FAS, and all the molecules that are required, that are expressed usually on short-lived effector cells. And the majority of the cells that expand during this time look more like MPEG, so memory precursor effector cells, and they express BCL2. And at the end of the contraction, most of the cells are actually central memory cells. And this is very weird because at the end of contraction, usually you still have effector cells and effector memory cells, and central memory cells are usually generated much later during the infection. But here at day 14, we have like 80% of the cells that look like central memory cells. So despite the fact that there are still lots of parasites present in the spleen, these cells seem to stay between day 14 and day 20 to not recognize any of the parasite. And at the same time, they get then reactivated at day 21, but instead of dividing and expanding again, what they do, they disappear. So we were interested first to know why they disappear. And we saw that actually they lose first their polyfunctionality, so the capacity to express IL-2 and TNF, and then interferon gamma, so they become dysfunctional, and they increasingly express exhaustion markers, so PD1 and LAC3 and TIM3. And so we were the first one in 2009 to describe exhaustion in T cells in a context of a parasitic infection. This was seen as well in other parasitic infection then later on. And what is interesting as well, that the mouse model is actually not that bad, because these cells, exhausted cells, were also seen in human visceral leishmaniasis by Susanne Len's group, and also in dogs by Christine Peterson's group. So can we revert this exhaustion? We know that dendritic cells upregulate PD-L1 or B7H1 over the course of the infection. So we blocked with the anti-PD-L1. And we can see that we can maintain the number of CD8 T cells instead of losing them over the course of infection. And this has an effect as well on the parasite burden, which is greatly decreased. So I know that people working with cutaneous leishmaniasis would now say, okay, PD1 is expressed by CD4 T cells as well, so it's not only CD8 T cells in this case. And I would say, yes, you're right. So that's why we have another way of proving that if you reactivate CD8 T cells, you can actually reduce the parasite burden. And this is what we did. We transferred the OT1 the day before infecting with amastigotes, transgenic amastigotes. And then what we do, we super infect at day 28, so when there is a chronic infection already established, with either vaccinia virus or vaccinia virus expresses Synfecal. So this virus will be recognized by the OT1 cells, and this one, the wild type virus, won't. Or we left them alone, and then we follow the expansion of CD8 T cells, and we can see that the OT1 actually get reactivated and recognize the vaccinia virus Synfecal. We can get back at day 6 interferon gamma production and then polyfunctionality by day 9, and this had a significant effect on the parasite burden. So we have a significant reduction in the parasite burden. So this was demonstrated actually that if you reactivate CD8 T cells, you can reduce the parasite burden. And I know that Paul Case has clinical trials now going on, working on a more refined approach than what we did in the mice to reduce parasite burden and by inducing CD8 T cells. Now, the next question we wanted to know is, okay, why they are not expanding during the early time points of infection. And then we thought that perhaps would be the problem is with the antigen-presenting cells, since there is a lot of IL-10 that is produced after 3 days of infection. It's possible that antigen-presenting cells are not capable of sustaining these responses. So what we did, we infected with ova-transgenic parasite at day 0, and then we transferred the cells this time. Instead of transferring the day before, we transferred 4 days after. And we transferred a limited amount of cell as well, at 20,000 cells at day 4. And then we followed those cells to see whether they would expand or not in the spleen. And to our surprise, this day 3 will be the equivalent of day 7 post-infection, day 11 post-infection, then 16 post-infection. And we can see that between day 7 and day 16 post-infection, the parasite, the CD8 T cells do not expand that much. And you see here an increase in numbers just because the spleen is enlarging. So it's like an homeostatic proliferation rather than an activation through antigens. And there is also hardly any interferon gamma that is produced. So this suggests that perhaps the CD8 T cells cannot see antigens because antigen is not presented. To exclude the possibility that clonal competition would play a role, we try to do the same with vaccinia virus. And then we also transferred the OT1 cells 4 days after infection with recombinant vaccinia virus expressing synfecal or wild-type vaccinia virus. And we can see actually that they expand, they undergo clonal expansion and clonal contraction. And they also, they get primed, so they produce interferon gamma, which is not the case with the wild-type virus as expected. So that means that despite the clonal competition, cells are capable of expanding, although to a lesser extent, but not in leishmania. So this has strong implication if you work on vaccines, because you can have the best T cell memory responses. But if antigen is not presented after 3 days, this doesn't bring much, because T cells, usually central memory cells, are stored in the secondary lymphoid organ, and they take about 3 days to be reactivated. And once they move to the site of infection, no antigen will be presented, so they cannot really be effective. And so the best would be either to have, like for leishmanization, effector cells circulating all the time, or like Phil Scott was showing, tissue resident memory cells, which will be activated straight away and they will be already in situ. So that's why lots of vaccine probably fail for leishmania because of this problem. So we have really to target T cells to a different site to get protection, or to get like a leishmanization effect. So is it that dendritic cells are not presenting antigen that we don't see that, or is the environment of the spleen that is suppressive? So to see that, what we did, we purified dendritic cells at various time points after infection, and then put them in culture with OT1 cells to see whether they would drive a response or not. And as you can see, it's not a fantastic response, but they can actually present antigen. So that means that ex vivo, in a very nutrient-rich environment and not absent, in the absence of IL-10, in the absence of hypoxia and inflammation, all dendritic cells would be capable of presenting antigen. But why they don't do it in vivo? So we blocked IL-10 and we could see an expansion as expected somehow, because this is suppressing the effector function of dendritic cells. But we also wanted to know whether there were other factors that contributed to that. And then we thought that probably hypoxia could be a problem, or the upregulation of HIF. So HIF is hypoxia-inducible factor 1 alpha. It's a transcription factor that is a key regulator of cellular response. Under normoxin condition, this transcription factor is degraded in the cytoplasm. And under hypoxic condition, actually, HIF is accumulating in the cytoplasm, and then it moves to the nucleus, HIF1 alpha, where it's dimerized, heterodimerized with the HIF1 beta to induce hypoxia response element containing genes in all the cells. So to help cells survive in this difficult environment that is an hypoxic environment. However, HIF can also be induced by under normoxin condition by pro-inflammatory cytokines such as TNF and IL-1 beta, and also by TLR ligation. And what is interesting, Leishmania promastigote as well as Toxoplasma, they actually can stabilize HIF by themselves. So it was shown for the first time in a group in Brazil, actually, that Leishmania induces HIF and requires HIF for surviving inside macrophages. And then an Indian group also described the mechanism by which Leishmania actually stabilizes HIF by interfering with the enzyme that degrades HIF. In any case, so that means that this parasite evolved to stabilize this factor. And when we wanted to look whether it's upregulated or not, and we see that it's upregulated increasingly in dendritic cells over the course of infection, not only by messenger RNA, but also by protein, we see that. And so we generated HIF CD11C specific HIF knockout mice to see what is the role of this transcription factor in the dendritic cells. And so we infected those mice and then purified dendritic cells at day 1, 3, 5, 7, and 14, and then look at the production of expression of IL-12, P35, and P14, also IL-10. And as was described in the literature, the Cre negative that correspond to the wild type mice, they actually lose the capacity to produce IL-12 during the early days of infection, whereas in the absence of HIF1 alpha, we can see that the expression of IL-12p35 is sustained, and we also got a reduction in the expression of IL-10. Does this result in increased priming of CD8 T cells? So we infected those mice, and before infecting, we transfer OT1 cells, and then infected with ovatransgenic parasites, and we could see that we got a greater expansion in those mice. So then at day 7 and at day 14, between 3 to 4 fold increase. And this was not only in the number of cells, but it had a higher frequency of short-lived effector cells and a slightly lower frequency of memory precursor effector cells. So that means that HIF1 alpha hinders the function of dendritic cells during the early stages of the infection. So this suggests that actually during experimental visceral leishmaniasis, CD8 T cells are kept in chains somehow. So in the beginning, they are hindered in their expansion capacity, and then later on they become exhausted. When they come into swing, there is inflammation, there is environment that could prime them, there is too much antigen, and they become exhausted. So it looks like in visceral leishmaniasis, the situation is exactly the opposite of what you see in mucocutaneous leishmaniasis, where CD8 T cells are overactivated and cause pathology. Here, CD8 T cells are completely locked down and they cannot do anything. So perhaps it would be better to have something in the middle to be able to have a proper function on these CD8 T cells. And it looks like Leishmania is using two strategies to actually hinder the CD8 T cell functions. So now to the CD4 T cells. And CD4 T cells in visceral leishmaniasis are firstly detectable at day 14, a very low level. And then by day 21, you start seeing higher frequencies. And by day 28, you have they are in full swing and they start dividing. And so this late priming could also be a consequence of the fact that during the early stages of infection, there isn't much antigen that is presented, and CD4 cells require a larger amount of antigen to be primed than CD8 cells. So that's why CD8 cells get primed at the beginning, and then this priming is not sustained, and CD4 cells, you don't see anything until really there is enough antigen, and the immune response actually changes, because there is this, how do you say, this disruption of the marginal zone, and also emergency and hematopoiesis that starts, and stronger inflammation that will start all of a sudden. The problem is that CD4 cells are primed, and during in an environment, it is actually a chronic inflammatory environment, and with all the consequences this entails, like strong anti-inflammatory responses, mainly IL-10, hypoxic environment. You have a lot of cells that are streaming into and moving into an organ that is highly vascularized, yet they, because there are a lot of cells, these consume more oxygen and then will create an hypoxic environment. And at the same time, you have a lot of tissue disruption that is going on because of the TNF. So how CD4 cell cope with all this in this environment, and how these factors influence the maintenance and development of these responses. So I'm going to go very quickly about the IL-10, because we have heard so many times that IL-10 is bad for the host immunity against leishmania. So it's one of the major regulators of the anti-inflammatory responses, and IL-10 knockout mice are highly resistant to Leishmania donovani. It's now 20 years that has been known that. And we have shown as well, when I was still in Paul Kay's lab, that the main producer of IL-10 in visceral leishmaniasis are FOXP3 negative CD4 cells co-producing IL-10 interferon gamma, and these cells are called TR1, and they were also seen in human. And there are also cells that can produce IL-10. We have seen dendritic cells, but also NK cells, and we have seen as well B cells. But we have shown in collaboration with Jay Bream at Johns Hopkins that T-cell derived IL-10 is the most important source of all. So if you deplete IL-10 in T cells, or if you have T cells that hardly express any IL-10, they behave more or less like IL-10 knockout mice. All right, so now hypoxia. We have seen that hypoxia affects CD8 cell priming during the early events. And now we want to know whether hypoxia could affect as well CD4 cell priming. And first of all, we looked whether the spleen was hypoxic or not. And we did the proxy probe. These are naive mice. And at the day 28, we see an increase of the staining. So this staining is very weak. So if you try to do it, don't be discouraged. It's not really a major shift. And we looked as well at the HIF expression, HIF1 alpha expression in all splenocytes. And we see that is peaking at day 21, at exactly when there is this emergency hematopoiesis, when it occurs. So you have a lot of monocytes and neutrophils and myeloid cells in general moving into the spleen. So the environment becomes hypoxic, but is significantly upregulated over the course of the infection and the chronic phase. Does it have an effect on the CD8 cell priming and DC function? So we infected our Cre positive and Cre negative HIF1 alpha CD11C specific knockouts, and then we purified the dendritic cells at day 14, 21, 28, and 35, and look for IL-12, B35, and 40, and IL-10. And we could see that, as expected, in the Cre negative, so which are HIF sufficient, there is hardly any IL-12 that's produced by dendritic cells during the chronic phase of infection. Whereas if you remove HIF1 alpha in CD11C positive cells, you can see that this IL-12 expression is sustained, and we hardly have any IL-10 expression. in dendritic cell. Does this result in increased priming of CD40 cells? Yes, it does. And we have a higher frequency of CD40 cells producing interferon gamma at day 28 and at day 35. Not only higher frequency, but the cells that produce interferon gamma also produce more interferon gamma. So also higher MFI for interferon gamma. So that means that HIF is really hindering the C function, really significantly hindering the C function and during the chronic phase as well, not only for the CD8 but for the CD4s. And what is interesting is if we remove HIF during the chronic phase, our CD8 become more exhausted. So that means there is even more antigen presentation probably occurring. But CD4s are better. Now, as I mentioned to you, we have during starting from day 14, there are the emergency hematopoiesis actually starts, and we have a lot of myeloid cells moving in, and we have among them is the inflammatory monocytes that we see, and alternative monocytes as well. So these inflammatory monocytes are very permissive. Both of them are very permissive to leishmania. And we have shown in collaboration with Krista Inonen that they express MSC class 2 and SCA. And these are markers that are been associated to regulatory monocytes. And these are already like this. I mean, they come out of the bone marrow already SCA and positive and MSC class 2 positive, and they are already very permissive in the bone marrow. And when they arrive to the spleen, they are also very permissive for infection. So like safe houses moving into the spleen. And so then we wanted to know whether HIF-1 alpha would play a role in these myeloid cells or not. And the mouse model that you should be using will be Lis-M-Cre mice, like has been described in the literature. So we generated Lis-M-Cre HIF-Flox, and we didn't see that there was a paper that suggested that actually for splenic myeloid cells, these mice hardly cut the antigen, the molecule you wanna use. So that is really, they are really not the right model to be using, but we generated them already. So we tested these mice, and we looked first whether they would ablate HIF in the myeloid cells. So we infected the mice, and then we purified CD11b positive cells. And then we looked at day 7, 14, 21, and 28 for the expression of HIF-1 alpha. And as you see, they behave exactly like wild-type mice. So this M-Cre shouldn't be used for splenic myeloid cells. It also doesn't cut in monocytes, in blood monocytes. I don't know in tissue, when they move to the tissue, whether this will be the case. But in the case of leishmania, it doesn't work at all. So please don't use that. And if you see papers that they describe the use of these mice for the spleen, ask them to show whether it works or not. So because most of the CD11b positive cells express CD11c, we used our CD11c knockouts, HIF-Flox knockouts, and we purified the myeloid cells, and we could see actually that there is a decrease in the HIF expression in these mice. So it's not a complete one because it doesn't work in these mice do not deplete neutrophils. Whereas the Lis-M-Cre are very good for neutrophils, even in the spleen, but not for monocytes and macrophages. But these work in monocytes and macrophages, but doesn't work in neutrophils. So there is a residual HIF in the spleen. So it's good to use them. So we went and we wanted to know whether this would affect the function of these monocytes. And usually you read a lot of literature for cancer because I see a lot of parallels between the environment in the spleen during chronic visceral leishmaniasis and cancer environment. And in cancer, they show that HIF-1 alpha actually enhances myeloid-derived suppressor cell-like effectors, functions. So we wanted to know, first of all, whether MDSCs are present during visceral leishmaniasis, during the chronic infection of VL in mice, and whether this cell's function is affected by HIV, the presence of HIV, the upregulation of HIV. So we infected the mice and then purified the CD11B cells at day 0, 14, and 28. We chose 14 because it's before the emergency hematopoiesis occurs, and day 28 is after the emergency hematopoiesis occurs. So this is a kind of a better control than day 0 somehow. And then we purify CD40 cells from a wild-type mouse and culture them under a Th1 condition, so it's anti-CD3, CD28, so it's not antigen specific, recombinant IL-12, and then we co-incubated these cells with CD11B positive cells from day 0, 14, 28, positive and Cre negative, so HIV sufficient and HIV deficient. And then we checked interferon gamma production. So with cells purified at day 14, we could see that they were somehow a bit inhibiting, about 10% inhibiting the interferon gamma compared with the cells purified at day 0. And but there was no difference between HIV sufficient and HIV deficient myeloid cells. When we did the same by incubating them with cells purified at day 28, we could see that actually in leishmania donovani there is a lot MDSCs are present, and these monocytes exert inhibitory function on the T cells, inhibiting Th1 responses, and this inhibition was somehow reduced by the half if HIV was absent. So that means that during VL as well, we have an function, the inhibitory function of DMCs is enhanced by the presence of HIV. So now we move to the third point. So we have seen that there is inhibitory responses, then IL-10, then we have hypoxia, the effect of hypoxia, and the third one will be tissue disruption and how this affects the Th1 development. And this occurs between day 14 and 21, that we start seeing a progressive disruption of the tissue, this explaining marker architecture. And before I show you some results, I want to talk about RF5, because it's really strictly related to what I'm going to show you. So RF5 is a transcription factor that stands for interferon regulatory factor 5. Has been described mainly in antigen-presenting cells, and in these cells is activated by TLR7 and TLR9 via MyD88 TRAF6, and then is induced in type 1 interferon and key pro-inflammatory cytokines such as TNF, IL-6, and IL-12. This transcription factor can also be directly induced by viral infection. Newcastle disease, for instance, virus, for instance, can do that. Also directly by type 1 interferon. So we wanted to know, when I was still at Hopkins, we were curious to know what this factor is doing since it is inducing inflammation, what it's doing during VL. And the first things we were set to do was to purify different cell subtypes and then see which cells were expressing RF5 over the course of infection. And then I asked my postdoc to add T cells as a negative control, because I wanted to be sure that the qPCRs were correct. And to our surprise, we saw that T cells were actually upregulating RF5 messenger RNA, but only during chronic infection, not during acute infection. So at day 7 and 14, we don't see that. And interestingly, we were working as well with TLR7 knockout mice at the time. This expression was totally TLR7 dependent. So these are total TLR7 knockouts. So we don't know how TLR7, what role it might play in upregulating RF5 in our CD40 cells. So when I moved to Canada, one of my students started a PhD project and trying to define the role of RF5 in the CD40 cells and confirm the fact that this RF5 is expressed during the chronic phase of the infection in CD40 cells and not before. We did it by fax as well. We saw that about 10 to 15% of the cells, depending on the infection, are positive for RF5, day 28. And because this is a transcription factor, so to be active, it needs to be in the nucleus, we did image stream and we look for the nuclear localization of this transcription factor that you can see here. These are two examples of nuclear localization of RF5. And these are two examples of a cytoplasmic localization of this transcription factor. So we could see that by day 28, about 80 to 90% of the cells had an active RF5 in CD40 cells. So that means it's doing something, but there was no literature at all about what this transcription factor is doing there. So we wanted to know first who is expressing it. So we saw that our effector and memory cells expressing it rather than naive. A bit is increasing over the course of infection, but is to much lower level. And the majority of the cells that are expressing it are actually gamma producing CD40 cells. And about 50% of the gamma producer express RF5 at day 28. So we were really curious to know what does it do in those cells. And we went on to construct T-cell specific RF5 knockouts using the LCK-3 and the RF5 flox. And then we infected those mice and looked at gamma producing CD40 cell frequencies. And we could see that actually they were increasing a lot. It was much higher in the absence of RF5 in T cells. So that means that somehow RF5 was inhibiting this cell. So we looked whether it was inhibiting gamma production and in vitro, and this was not the case. So we thought perhaps this is killing the cells or doing something to kill the cells. But there was no literature about CD40 cells dying during leishmania. So first we wanted to know whether they do die or not. And to do that, we use a very simple adopted transfer experiment. So we use congenic mice, CD45.1 mice, and then we transfer bulk CD40 cells purified from the spleen of wild-type donors. And we follow those cells. We transfer about 10 million cells, and we follow those cells over the course of the infection. And we could see that after day 21, we could start seeing this population here. So there are like CD40 cells down-regulating CD40 cells. So first I thought that my students didn't purify properly the cells and were contaminants and give it to somebody else to do at the same time, and everybody was getting the same results. And then we started seeing, okay, what are these cells? And they are 20% of them still express CD3, 30% of them are annexing positive, and about 70% of these cells are B220 positive. So B220 is not only a marker for B cells, but on T cells, B220 is a marker for apoptotic cells. So that means that the CD40 cells massively die starting from day 21 on, because you can see the percentage here is much higher than the percentage of cells that are alive. So we were really surprised to know that. And we thought whether we wanted to know whether RF5 has something to do with that or not. And interestingly, if you can see this are wild type, if we transfer wild type CD40 cells, so Cre negative CD40 cells, we have still this population appearing, but when we transfer RF5 knockout CD40 cells, we don't have that. So, and when we infect our Cre negative and Cre positive cells and look for annexin 5 expression in CD40 cells, we see that actually in black you have the Cre negative, so the RF5 sufficient, and you can see that they increasingly express annexin 5 over the course of infection, which is not the case for the Cre positive cells. So that means that RF5 has something to do with cell death in Th1 cells, in gamma producing Th1 cells. All right, so we knew that TLR7 has something to do with the expression of RF5 in CD40 cells from previous work, but was done with in total TLR7 knockout. So we didn't know who is doing where is required, and we thought that most plausibly that is expressed by TLR by an antigen presenting cells, and this will then in parasite will be recognized by TLR7 and then induce type 1 interferon, which then will induce RF5 and kill CD40 cells. And alternatively, we thought that unlikely TLR7 is expressed by T cells, and then this will somehow induce RF5 and kill by whatever mechanism, we don't know, T cells, and we don't know what is triggering TLR7 either. So to cut the long story short, because I don't have time, this first more plausible explanation is not true, is not via type 1 interferon, and the second one is true. TLR7 needs to be expressed by T cells, and I'm going to show you why, because if you adoptively transfer TLR7 knockout CD40 cells and wild type CD40 cells, you can see in the wild type CD40 cells upregulate RF5, but if you transfer TLR7 knockout T cells, you don't get an upregulation of RF5 or just a minimum one. So this means that TLR7 is upstream of RF5 in T cells. And what is downstream of RF5? So we went back to the literature and look at less known targets, and again back to cancer biology. And we found that death receptor 5, also called trail receptor 2, is one of the downstream targets of RF5, FAS as well, and TNF receptor as well. And that RF5 can somehow control cell cycle, can induce cell cycle arrest and cell death in tumor cells as well. So we were really intrigued to know whether TLR5 is upregulated on our cells, and it is, on CD40 cells is upregulated, and more so at day 28. And we wanted to know whether TLR7 is expressed on our CD40 cells. So we purify them from infected animals day 14, 21, and 28. And we see that at day 14 is hardly expressed, but then by day 21 and then 28, it starts being upregulated. And this upregulation of TLR7 on CD40 cells was also reported in HIV and in other chronic infection and hepatitis C, I think, as well. So it's not only in leishmania, and only during chronic infection it comes up. So we wanted then to know what is upstream. Is it really TLR7, and what triggers TLR7, and what is downstream? For that, we infected our mice, and then we sacrificed them at day 14, 21, and 28. We purified CD40 cells, and then we stimulated our CD40 cells with various TLR ligands, like imiquimod for TLR7, CpG for TLR9, LPS for TLR4. We wanted to know whether they are capable of microbial sensing. So we use live and dead parasites. We use parasite RNA, parasite DNA. And we wanted to know whether they, since there is a lot of tissue disruption, we wanted to know whether they are able of sensing damage-associated molecular pattern. So we use supernatant from apoptotic cells. Our readout was RF5, DR5, and cell death using annexin-5 and propidium iodide. So here on the left, you have the results for RF5, on the right for DR5. The first graph here are the cells purified at day 14, so before TLR7 is expressed. And these are cells that are purified at day 21, when TLR7 is already expressed. So we hit them with all these stimuli. And as you can see, the only two that actually induced RF5 expression were imiquimod and the supernatant of apoptotic cells. But these two didn't have any effect on cells at day 14, purified at day 14, because if you remember, these cells do not express TLR7, and also during the infection do not express RF5 at the time. So that means that starting from day 21, if you trigger TLR7, or you trigger with imiquimod, or if you expose the cells to supernatant of apoptotic cells, you can induce RF5. And this went along as well with the upregulation of DR5. Can we have cell death? So cell death was there with imiquimod, and was there as well with supernatant. So that means that we could see the axis TLR7 inducing RF5 that went then inducing DR5. So, but then we wanted to know if supernatant also goes via TLR7 or not. So if it's TLR7 responsible for recognizing this dump. And we performed the same experiment. These are cells purified at day 14. And non-dead sites are cells purified at day 28. And then we use an inhibitor for TLR7, an antagonist. And again, cells at day 14, we didn't see any difference in any upregulation of RF5. This time we did it by qPCR. And but when we add imiquimod, we have an upregulation of RF5, and with supernatant an upregulation of RF5, as I've shown you before. If we put the antagonist of TLR7, we abrogate this upregulation. So that means that the supernatant triggers TLR7. So the dump recognized by TLR7 expressed on T cells. We wanted to know whether what is really triggering this TLR7, and we couldn't find what it is, because it's not pure supernatant purified from the apoptotic cell material and or pure DNA. So we don't know what it is. So to summarize this, we could see that T cells during chronic infection are capable of sensing tissue disruption, to sense dump via TLR7. This will induce the upregulation of the RF5, which then activates and then move to the nucleus and drive the transcription of DR5, also caspase 8, I didn't show you in the interest of time. And this will make the cells more prone to trail-mediated apoptosis and cell death. All right, so it looks like inflammation is causing a lot of damage in leishmania. This type of inflammation that leishmania develops is causing a lot of damage, and would be interesting to know what is actually initiating and maintaining these inflammatory responses. And here I'm coming with something that is a bit speculative, but you will see that perhaps is correct or perhaps not. I'm open for discussion. So we infected AID knockout mice, and these AID mice are incapable of producing hypermutated and or class switched antibodies. And so they do not develop hypergammaglobulinemia, and they are not capable of producing antibody against the parasites. So we infected these mice with leishmania donovani, and then compared the expression of different cytokines in the spleen, TNF, interferon beta, and IL-10. With wild-type mice infected with leishmania as well. And we could see that the kinetic of expression of this cytokine is inverted. I'm sorry for the colors, they're very, very similar. One is dark gray and the other is black. So dark gray is the AID knockout, so those that they cannot produce antibodies, and the wild-type are in black. And you can see that the ID knockout are much more inflammatory right at the beginning of the infection, but then somehow they are not anymore. And the wild-type mice are exactly the contrary. So it's like an inverted kinetic. So this goes along with the fact that antibodies were described by David Moser's group to induce IL-10 in macrophages. And you can see that during the early stages of the infection, actually they do so, because in the absence of antibody, you have a more pro-inflammatory environment. However, when the hypergammaglobulinemia starts, is probably that antibodies do exactly the contrary. They induce inflammation, and will be more like in autoimmune diseases, like in arthritis, for instance, where antibodies are responsible for inducing inflammation. So it's like it's an amount, it's a question of amount of antibodies that is making the difference. And so interestingly, in this AIG knockout mice, we have also better CD4 T cell responses producing interferon gamma, and we hardly have any TR1 cells. So this double producer gamma IL-10 are hardly developed in these mice. So it looks like really these antibodies are really generating a certain type of inflammation that is detrimental to the immunity of the host. And of course, we have a reduction in the parasite burden. So instead of increasing, and here is the day 21 is when there is the onset of this hypergammaglobulinemia in mice, it's just decreasing. So we came up with, to summarize this part of the CD40 cells with a model that I'm open to discuss. Where we see that the spleen goes from this size to this size between day 0 and day 28, and between day 14 and 21. We have a disruption of splenic microarchitecture. So the structures, the marginal zone is no longer maintained, and B cell area now are more in contact with with infected macrophages, which could cause perhaps that the polyclonal B cell activation and with consequence of hypergammaglobulinemia. And sorry, and this will exacerbate inflammation. We have an increase of TNF and type 1 interferon especially, and this will cause this exacerbation of inflammation, will cause an hypoxic environment with an upregulated HIF-1 alpha, which inhibits DC function and enhances MDSC's function, will also call in regulatory responses, the IL-10 gamma double producers, the TR1s, and at the same time will also cause T cell disruption, which then regulates RF5 is sensed by TLR7 by Th1 cells, and this will induce RF5 upregulation and the cell death. So all these mechanisms in light blue are mechanisms that are there to protect the tissue. And if you block them to improve immunity, we still have this problem that is the inflammation going on. So what I think we should do more as an immunotherapy is to block inflammation, either try to block this polyclonal B cell activation or try to block what is causing this inflammation. And I think type 1 interferon is one of the good targets that could be used to improve immunity against VL. In cutaneous leishmaniasis, you have more IL-1. This will be the correspondent of IL-1 in visceral leishmaniasis. IL-1 is not so highly expressed during chronic infection, is more type 1 interferon. Okay, I would like to thank all the people that did this work. So the current and past member of the work of the lab, especially Akil that did all the CD8 work, Sasha did all the B cell work, Emeric did the CD40 cell work with Tanya, and Lynn is still here, and Sasha was there as well. Now she left, and we did it in collaboration with Krista Inonen, that is working on the bone marrow, Julien van Grevenenge, and all this work on IRF5 wouldn't have started if it wasn't for Paula Pita. She's unfortunately no more with us. Was a fantastic scientist, and she approached me to ask me whether I wanted to know what is IRF5 doing in my infection model, and everything started because of her. She was at Johns Hopkins, and Andrea now is working with David Sachs, and Jay Bream at Johns Hopkins as well for the collaboration. So, and of course, all the funding agencies. Thank you very much. If you have any question, I'm more than happy to take them. So let's stop sharing.
Yes, please, Simona.
All right.
Simona, fantastic, your presentation, very, very
was a bit too
impressive.
long.
No, it was beautiful because you showed a story, you know, that you were following for a couple of years, several years actually, and you can see that you have several points that you can interfere to control the disease, but they are very difficult to the points that you can interfere. It's very difficult to do that. For example, hypergammaglobulinemia, how you can do that to stop the production of these molecules.
Well, we need to investigate.
That's complicated.
Yeah. We need to investigate more in that direction. That's the fact. And are there no one doing anything?
Yeah. So CD8 cells, they are not the focus anymore for the control of the disease. If I well understand, I'm not an immunologist, but if I well understood, the CD40 cell, no?
No, CD8 would be. They are not. They are kept. They are kept in chain. So we have to. But now there is already
the good moment.
Yeah, but Paul is doing a clinical trial. It's no point to do anything with the mouse if somebody's doing things with human. You know, we do stuff with the mice to understand to go then to human. Absolutely. But if there is already,
fantastic. We have several people that are congratulating your presentation. Some have to go to other meetings, Claudia Brodsky and Manuel Barral, people that are very interested in the field. And Camila, she's asking here, would this mean that, I don't know in which point she asked that, but would this mean that VL, CD8 cells, would be the main effector population rather than the CD40 cells? And the parasites actively restraining this? Also, she finalized. Also, I may have missed this, but this restraining CD8 cells is similar in spleen and liver?
No. Okay, so it's not similar. This restraining of CD8 cells is in the spleen, but not in the liver. In the at the beginning, yes, the expansion of CD8 cells is restrained, but in the liver, the CD8 cells are not becoming exhausted. So they are fully capable and fully functional. So the same is for the CD40 cells. CD40 cells are also not becoming dysfunctional in the liver. They are really in swing and fully capable, but not in the spleen. So that means that there is the environment of the spleen that is really suppressing all of these responses. And you have to take into account that the liver is an effector organ, the spleen and a secondary lymphoid organ, additionally to being an effector organ. You know, so it's a site of infection. So CD8 cells are actually not, they are non-existent. So if you look at them, they hardly do anything ever in any space, in any place. So very, very, very little a later time point, but not in the liver.
Okay, Taina Luiz de Souza, she has excellent talk, Dr. Simona Stäger. Do you think that this reduction of cells in the spleen, CD8 cells, may be related to cell exhaustion and apoptosis, since there are studies showing that cell exhaustion can trigger apoptosis?
Yes, it is what I meant, what I said, that it was T-cell exhaustion, and then the cells were dying because of apoptosis. The CD8. CD4, no. Because we looked for RF5, if it meant the CD4, then not the case, because they die before they become exhausted. CD4 become exhausted after day 28, you start seeing a functional exhaustion, not so much before, while we see they dying at day 21. So it's inflammation induced cell death, and it's very different than exhaustion. And RF5 is not expressed on exhausted cells, if this was the question. But for CD8, yes, they are dying because of the exhaustion.
Great talk, Juliana and Marilda Gonçalves, Celis. Very interesting data, congratulations. I may have missed the explanation. The effector memory cells would be more prone to death mediated by IR5. They would express toll-like receptor 7 in higher amounts.
Wait, I might have this.
Celis.
Effector memory cell would be more prone to death mediated by RF5. Yes, we have. We don't know whether they express more TLR7 or not. It's possible. What we think is possible, because we didn't sort them. So we just look at TLR7 in the whole CD4 population, because they are really too little to sort them somehow, a small amount. We could do it, but we should have a pool of really a lot of mice. And is it possible that TLR7 is upregulated? Because I think it has something to do with TCR engagement. And we are now looking what is inducing the TLR7 upregulation, because it would be very interesting to understand. And why is it during chronic infection that is coming up and not during acute infections? Because these mechanisms are not operating in acute infections. So, and those Th1 are more prone to cell death. We don't see the same for naive cells. We did the same. We are now doing a study with HIV with a colleague, and we see exactly the same. But this time we divide it into memory and effector, and then naive cells do not express TLR7 and they don't die via RF5.
Mittermeier, nice, Felipe, nice presentation. Yes. Do you think that different immunoglobulin isotypes can influence the response profile?
This is something that I would love to know. I don't have the money for that, and but it's something that I would love to know. But it's something that should be done in human, actually, because in mice it doesn't make much sense to do a work like that. So it would be good to look at the human response and then check whether different isotypes and level of the different isotypes and the type of responses you have. That could be something that could be very interesting to do.
Washington dos Santos, works a lot with spleen in dogs infected with naturally infected with leishmania. Excellent insightful conference. Does the lymphoid follicles tend to disappear in these animals?
Yes. So the B cell, yeah, the B cell follicle, they shrink completely, and the number of B cells also disappears. You have more plasma cells, but the follicle disappears. So you antigen-specific production of antibodies actually is decreasing after a certain moment in these mice. But you have hypergammaglobulinemia that is well, we published this maintained by type 1 interferon, but there are other things that are still going on, I guess. It's actually, these would be interesting to know what type of antibodies are those, and the specificity and the affinity, and we don't know much about that.
Simona, you showed the connection between spleen damage and bone marrow, but do you have actually, regarding the HIF-1 alpha and then also the activation of the cells that are more prompt to be infected with leishmania in bone marrow cells? But you know what is going on between spleen and bone marrow as in a time course when the cells CD8 cells they disappear, no? And what happened in the bone marrow at the same time?
I'm ashamed to say that the bone marrow was completely neglected. Even more than the B cells, was completely neglected by everybody. So I know that Paul Kay did one paper in 2000.
He did one paper several years ago, I think.
Yeah,
20 years,
yeah, exactly.
Maybe.
But otherwise, the bone marrow was completely neglected by everybody. Yeah. So the time course in the bone marrow, unfortunately,
that's
because
interesting,
I think
because the mouse models, they're interesting to look at.
Yeah, and especially the parasite is in the bone marrow. So
yeah.
It would be interesting to see what it's doing. I know that Krista is looking at that, so I cannot say, I cannot talk for her results and her stuff, but it's but I think it's a very important point to look at, and your work with the dogs would be also very hot.
Yeah. Washington, he answered you. Interesting. We have observed this decrease in the density of leishmania specific plasma cells in the spleens of dogs as well. The naturally infected dogs, where he showed the disorganization and the spleen disorganization
but in the spleen
severe infected dogs.
Yeah, in the spleen is somehow expected because as soon as you do an emergency hematopoiesis starts, it will push away all the rest of the production. This is what Krista told me. That is typical. That is decreasing.
Yeah. And these alterations are probably responsible for disorganization of the spleen, probably in dogs as well. Yeah.
Yeah, but they follow the disorganization. That's the problem.
Yeah.
You know, I don't know what is the chicken and the egg, you know, in that case.
Yeah.
I know that trypanosoma does the same. Trypanosoma also kills B cells.
And did someone try to treat these mice and the model that blocks the HIF-1 alpha, or do you think that it's too late and to reduce the HIF-1 alpha effect?
The HIF-1 alpha is a very difficult molecule to target because it's strictly required in the bone marrow, for instance, because it's physiologically hypoxic. So if you remove it, that's why total HIF-1 alpha knockout are dead. So it's not something I would touch. There are other people that in cutaneous leishmaniasis that say that is actually HIF-1 alpha is pro-host and not anti-host. And there is one paper that says that actually using the LISM-CRE that says that in visceral leishmaniasis as well is pro-host and is good. I just need to mention this controversy, but I don't believe those results because we use the LISM-CRE and it doesn't delete HIF. And I don't understand how a parasite evolved to how do you say, evolved to stabilize a factor that actually is required to kill it. So it doesn't make any sense to me. So, but if an alpha I wouldn't touch is at the target as such. It would be better to look more at the downstream effects, and it's too difficult to touch.
Yeah. Okay, but it's a very nice talk and
but
really
type 1 interference is feasible.
Yeah. Yeah. No, because we can talk later, but but we several years ago we did proteomic studies in Leishmania amazonensis and Leishmania major infected cells, and we both found that HIF-1 alpha is upregulated in these cells related to uninfected, and that's interesting because it appears as well in Leishmania infected cells, both amazonensis and major infected animals or cells in vitro. Okay.
Well, the first people that the first group that look at HIF-1 alpha was a group in Brazil, actually, several years ago.
I know. Yeah, I followed this.
Yeah. These are the little pearls that nobody ever looked at. So,
yeah. So thank you very much. I'm very happy to have you here, and it was a fantastic presentation. Very solid work in Leishmania and immunology, and we thank you very much for doing that.
Thank you for inviting me, and thank you for everybody to listen to the talk until the end.